Welcome, NEET aspirants. This question is from the chapter Cell, the Unit of Life. Let's attempt the question now. Statement 1 says concentrically arranged cisternae of Golgi complex. So, sacs of Golgi complex are called cisternae. This is correct. And they are concentrically arranged. This is correct. Are arranged near the nucleus. Correct. With a distinct convex cis. Yes, cis is convex shaped or maturing phase. Maturing is incorrect. Cis is forming phase, and because of this, the statement becomes incorrect. Statement 2 says a number of proteins are modified. Correct. Proteins are modified in the cisternae of Golgi complex. Correct. Before they are released from cis phase. Cis is again incorrect. This is trans phase. So both the statements are incorrect. Both the statements are false. Option number 4 is the answer. Mark it on the OMR sheet. 192 is 4. So see, you have to read the statement carefully, word by word, and analyze it whether it is correct or incorrect. And then you mark your final answer on the OMR sheet. Do not hurry in attempting two statement questions. So let's understand what Golgi complex is. Okay, what are cisternae? Then what is convex or the cis phase? What is trans phase? And overall, what is the role of Golgi complex in the cell? So Golgi apparatus was first reported by a physician, Italian physician Camillo Golgi in the year 1898. And he reported of observing a reticular material. So reticular means a network of sacs which are called cisternae in the cell. So the cisternae are flat disc shaped sacs. They will look something like this. Okay, then another sac. There's a network, so you will have several layers of these sacs. The Golgi cisternae are concentrically arranged near the nucleus with a distinct convex cis or the forming phase and concave trans or maturing phase. So the cisternae, which is close to, here is the nucleus, and then there is endoplasmic reticulum, RER. Okay, so the phase which will receive the newly formed protein, okay, the new vesicle will come and pinch in this area, it will merge in this area. So this is the reason why it will have a convex shape. It will look like this, convex shaped. And this is called the forming phase. It is convex shaped and it is called cis phase or forming phase. Now, this end, after protein modification, like glycosylation, the protein will leave the Golgi apparatus. So, this end, this cisternae, it will have a vesicle which will pinch out. Okay. So, in the process of pinching out, this end will extend little bit. And this is the reason it will acquire a concave shape. So the trans phase is concave in shape, and this is called the maturing phase of Golgi. So the flat disc shaped sacs are called cisternae. Don't get confused with another term which is called cristae. Cristae is the term of the inner membrane of mitochondria. So Christ is God, and Christe, God gives you power, and Christe, the inner membrane, is the source of power. The ATP generation will happen only because of the inner membrane. So that way you can remember. Okay, these are the two terms you might get confused. Okay, they sound similar, cisternae and Christe. So Golgi is associated with cisternae and mitochondria is associated with cristae. Then cis and trans phases of organelle are entirely different but interconnected. So the sacs which I drew here, so This way they will be connected. So another connection, okay? Then maybe this one will be connected from here. So this is how the sacs will, they are interconnected, okay? And then the protein transfer will be easy through these cisternae. The important point is they are interconnected. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of Golgi apparatus before they are released from the trans phase. Golgi apparatus is important site of formation of glycoproteins. So after glycosylation, the protein is called glycoprotein. And then glycosylation of lipid, it is called glycolipid. So you will have to remember these two names as example of biomolecules that are processed in Golgi. So let us visualize what we have read now. So nucleus, this has DNA. Okay, we are talking about proteins. So proteins are encoded in the gene. So by the process of transcription, the mRNA will be produced. Translation on the ribosomes of RER, rough endoplasmic reticulum. The convex shaped sac, which is called the forming phase of Golgi, it will receive the newly formed protein for processing. That is called post translational modification. The term is not mentioned in NCRT. You have to simply remember modification of the protein happens in Golgi. And then the trans phase, the concave shaped phase, or which is called the maturing phase of Golgi, it will receive the modified protein, and then these will be packed in vesicles, and by the process of exocytosis, those proteins will be delivered outside the cell. And then there are other vesicles, okay, that will carry the modified protein to other intracellular compartments. So Golgi plays the role of modifying the protein and then packaging it either to be secreted out of the cell or to be delivered to another compartment, intracellular organelle. So the two phase which you have to remember is cis. Cis is convex shaped and it is the forming phase. The maturing phase is concave shaped and it is called trans phase. The two different molecules which are given in example are glycoproteins and glycolipids. So now if you attempt this question, concentrically arranged cisternae of Golgi complex are arranged near the nucleus with distinct convex cis or the forming and concave trans or the maturing phase. That would be the correct statement. And statement 2, a number of proteins are modified in the cisternae of Golgi complex before they are released from the trans phase. So this is all about this question. Thank you so much for watching my video.